Good morning. This presentation will discuss the use of recombinant proteins in medicine, specifically the history and development of this relatively modern technique, how it works, full current applications, advantages and shortcomings, as well as potential future applications. Protein is a component of all organisms. A protein molecule contains one or more polypeptide chains made of amino acids, of which there are 20 naturally occurring. Amino acids are linked together by peptide bonds to form proteins, each with a unique sequence of amino acids. Protein function is completely dependent on their structure. Proteins perform enzyme catalysis, defense, transport, support, motion, and regulation. Proteins are essential in a highly complex physiological network, performing many precise functions. In the past, we obtained protein by isolating it naturally, an inefficient and time-consuming process. Recombinant molecule techniques have made it possible to clone the DNA, encoding a specific protein into a vehicle known as an expression vector, and express the protein in bacteria, yeast, insect cells, and mammalian cell hosts. Recombinant proteins are translated products of exogenous DNA in living cells. Recombinant proteins are made with two major steps: molecular cloning and protein expression. Recombinant proteins are currently one of the most powerful techniques used in biology and have a wide application in medicine, research, and biotechnology. Here I have included a timeline of significant events relating to the development of the technique. DNA technology really began in 1953 with the discovery of the double helix by Watson and Crick. Some of the first recombinant proteins used in clinical practice were somatostatin in 1976, insulin in 82, erythropoietin and interferon in 86, tissue plasminogen activator the following year, and coagulation factors in 1990. With this, biotechnology companies were created, and biotechnology industry organizations were founded to support the advancement of this new sector, and investment increased. It is estimated that these new products will represent more than 25% of the global pharmaceutical market in the long term. For context, by 2014, the global pharmaceutical industry eclipsed $1 trillion in annual revenue. Many recombinant proteins today are part of standard therapies, such as insulin for diabetics, vaccines for influenza, and the treatments of hepatitis A and B, cystic fibrosis, hemophilia, and in cancer treatments. I have included a list of some recombinant proteins and their clinical uses in this graphic. Some of them have been used in clinical practice for more than 20 years. Looking toward the future, some recombinant proteins in preliminary development may be useful in the treatment of multiple sclerosis, myasthenia gravis, scleroderma, phenylketonuria, and galactosidemia. The future of recombinant protein technology will be discussed in detail in future slides. Recombinant DNA result from the use of molecular cloning in the lab. It is a technique designed to bring together genetic material from more than one source. This is possible as all the DNA from all organisms share the same chemical structure, differing only in the sequence of nucleotides. Proteins that result from the expression of recombinant DNA are called recombinant proteins. Two classes of enzyme are critical in recombinant DNA technology: restriction endonucleases, whose job it is to recognize and cleave DNA at specific base sequences, and DNA ligases, which join or link fragments of DNA molecules together. Specialized vectors are required to necessitate restructuring of the foreign coding sequences. I have included here a Lehninger schematic, a simplified illustration showing the first 4 processes of DNA cloning. You can see here the cloning vector or plasmid in yellow and a eukaryotic chromosome being separately cleaved with the same restriction endonuclease. This cleaving isolates the DNA fragment of interest. The resulting fragments are ligated to the vector and introduced to a host cell for cloning, also called propagating, which produces many copies of the recombinant DNA. Imagine this process using insulin as an example. Insulin is a protein acting as a hormone to help us control our blood glucose levels. First, we need to locate and cleave the insulin gene from our very long genome. The restriction endonuclease enzyme does this. We then need to add this native insulin or protein to a vehicle called a plasmid. These are often taken from E. coli. Ligation occurs, whereby the fragments of insulin are brought together in the vector. The resulting product is known as a recombinant DNA molecule. A key aspect of this to remember is that the plasmids used here are able to self-replicate once it is inserted into the genome of a cell, commonly an E. coli cell. After replication takes place, transcription will too, and messenger RNA is produced. Translation into proteins will then occur, resulting in many proteins, or recombinant proteins. In this instance, insulin. Recombinant proteins are used in many research fields now, including molecular biology, cell biology, and biochemistry. They are used in structural and biophysical studies to identify and locate the position of the protein encoded by a specific gene, and to uncover the function of other genes in cell signaling, metabolism, growth, replication and death, transcription, translation, and protein modification. They are also useful tools in understanding protein-protein interactions and can be used to develop enzymatic assays as standards, for example the ELISA standards, and they can be used as positive controls in Western blots. Recombinant proteins are used in biotechnology, food production, agriculture, and bioengineering. For example, the breeding industry uses enzymes to add to animal feed to increase the nutritional value of the food, reduce feed cost, and support gut health. I will now discuss two current applications of this technique, insulin for diabetes and vaccines for influenza. 422 million adults have been diagnosed with diabetes worldwide. At least 85 to 90% of these cases are type 2, with an economic burden of $825 billion per year. For more than 50 years, we treated diabetics with animal insulin. Recombinant DNA technology made human insulin available in 1982. During the 80s and 90s, insulin analogues were created through manipulating the structure of the native protein. The very first clinically available insulin analogue, Lispro, proved that scientists could achieve improved glycemic control without an increase in hypoglycemia. Rapid acceleration of the basic and clinical research in this area, with the introduction of new short and long-acting analogues, increased stability and less variability, may provide individualized treatment opportunities in the future. Influenza is the most common infectious disease. The World Health Organization states that up to 30% of children and 10% of adults are affected with influenza annually. The severe complications caused by influenza result in the death of up to 500,000 people annually and an economic burden of $3 billion. Modern influenza vaccines generally include the formation of antibodies against influenza surface antigens, hemagglutinin and neuraminidase. The surface proteins undergo progressive antigenic variation, or drift, requiring annual renewal of the strain composition. The problems associated with having to attenuate pathogenic strains of the virus can be solved using recombinant subunit vaccines. The ion channel forming protein M2, as opposed to the two I previously mentioned, is largely considered the most promising candidate for influenza subunit vaccines. My colleague Trish will now take over to discuss the way in which recombinant proteins are currently being used in the treatment of cystic fibrosis and hepatitis B. Trish will also discuss the advantages of recombinant protein technology, as well as the shortcomings of the technique and potential future applications. Thank you.
Thanks, Laura, and hi everyone. My name is Trish, and I'll be continuing with two more examples of recombinant protein application. Scientists have utilized recombinant protein in the treatment of the condition cystic fibrosis, which is one of the most common genetic disorders. While the frequency varies worldwide, 1 in 2,500 newborns are affected in Northern Europe. Cystic fibrosis sufferers are defective in a gene that codes for a 1,480 amino acid transmembrane glycosylated polypeptide, which functions as a chloride channel, seen here in Figure 5. The major clinical finding of cystic fibrosis is excessive mucus in the respiratory tract, resulting in difficulty breathing. The excess mucus is caused by the defective protein, creating an imbalance in ion movement. The result is movement of sodium in the reverse direction, resulting in water retention away from the mucus layer. The mucus layer is therefore left thick and inelastic, indicated in Figure 5. Further, inhaled pathogens become trapped in the mucus, triggering an inflammatory response, which degrades the bacteria's DNA, leading to further accumulation in the mucus. DNase 1, a restrictive enzyme, is responsible for degrading extracellular DNA, reducing mucus viscosity. Recombinant protein technology has successfully produced synthetic DNase 1 for the use in cystic fibrosis. Through isolating cloned DNA and expressing it in Chinese hamster ovary cell lines. The synthetic protein is administered to the sufferer to aid in additional debris removal of the mucus and improves the quality of life. Recombinant proteins are also used to guard against hepatitis B. Globally, there are 2 billion individuals infected with hepatitis B virus, which targets human liver cells, while 1 million people die from complications each year. In the past, prophylactic treatment utilized in vivo hepatitis B antigens from the blood, that is, extraction of hepatitis B surface antigens purified from live donors. This technique was restricted by limited availability of infected human plasma and the risk of contracting intact viruses from blood donors. Current recombinant protein vaccines express the hepatitis B surface antigen in yeast cells, which are capable of producing highly functional protein, resulting in high efficacy and achieving immunization without the previous risks involved. Both advantages and shortcomings need to be kept in mind for a technique with such large-scale application. Both are discussed extensively in the literature, which holds great importance in the refinement and future applications of the technique. Some advantages include bypassing issues of source availability. With the development of synthetic protein, there is decreased demand or reliance on existing tissue sources for the extraction of proteins. It also overcomes the issue of source safety, as detailed in the hepatitis B example previously. It also has the ability to efficiently isolate functional proteins that are well defined. Additionally, the proteins can be expressed in large quantities, enabling large-scale research, leading to progressive discoveries as well as wide application in medical, industrial, and agricultural fields. Major medical advances have been achieved through recombinant protein development. As mentioned earlier, issues of source safety have been overcome. An example of this is the treatment of the blood clotting disorder haemophilia. Treatment in the early 1970s involved the delivery of the intravenous plasma-derived protein clotting factors. By the 1980s, it was tragically discovered that bloodborne viruses, HIV and hepatitis C, had been transmitted to a large percentage of haemophiliacs through contaminated plasma derivatives. Recombinant protein has eliminated some of those risks through the development of synthetic controlled techniques, producing clotting factors such as recombinant clotting factor 8 for haemophilia A. The development of vaccines has also been propagated. A recent example is in the field of oncology, where recombinant proteins in the form of antigens that represent surface tumor proteins are used to successfully stimulate the immune system, producing cytotoxic T lymphocytes, which are able to directly target malignant cells. There are also shortcomings associated with recombinant proteins, as each protein poses a new problem. That is, high levels of synthesis and stability are required to be optimized in each single case by empirical analysis of various parameters. To achieve this, time-consuming processes are required to establish the correct host for certain proteins. An inadequate host or expression system could result in poor growth of the protein, inclusion body formation, which occurs when synthesis occurs too fast, resulting in insoluble fragments that in turn affect the folding and the tertiary structure of a protein, and sometimes complete protein inactivity, as well as incorrect disulfide bond formation, which for many proteins is vital in achieving the biologically active 3D structure. There can also be formation of toxins, as well as inadequate post-translational modifications. For example, bacterial hosts do not have the machinery to glycosylate proteins well. Some other shortcomings associated with recombinant protein include the potential for misuse. Recombinant DNA technology has enabled production of synthetic glycoprotein hormone erythropoietin, or EPO, for therapeutic use in individuals with anemia associated with renal failure and cancer. Restoring EPO levels with recombinant human EPO allows adequate erythrocyte production and in turn oxygen supply. Unfortunately, this development has also facilitated the illicit use of EPO by athletes to improve performance in endurance sports. Additionally, the expense of the technology can also pose setbacks. With the need to continue improving safety and quantity of production, rigorous and lengthy trials are required along with protocols and administration. This introduces a cost barrier, and clinicians are forced to use outdated, less effective methods. Developments in the use of recombinant proteins have occurred at a fast pace and are likely to continue in the future. Current research revolves around new expression systems in the hope of producing larger, more complex proteins and continuing to improve the quality and the quantity of synthetic protein. Studies postulate that a third generation of recombinant proteins will aim to be completely free of animal or human protein. And there is ongoing pursuit in the ability to apply recombinant proteins in therapeutics via exact routes, such that the body delivers its own protein. Additionally, the field of oncology in particular is currently actively pursuing the ability to use recombinant proteins to limit drug toxicity while also enhancing drug targeting. Thank you for listening to our presentation, and here is a list of our references, which contains a mixture of peer-reviewed articles and textbooks. Thank you.